Well, it's my pleasure to introduce Dr. Sheena Josselyn, who's a senior scientist at the Hospital for Sick Children, for her talk entitled Making, Breaking, and Linking Memories. Dr. Josselyn is a senior scientist in the Neurosciences and Mental Health program at the Hospital for Sick Children and a professor in the departments of psychology and physiology and the Institute of Medical Sciences at the University of Toronto in Canada. She holds a Canada Research Chair in Molecular and Cellular Cognition and is the EJLB Scholar. Her program of research is dedicated to understanding the neural basis of cognitive function and dysfunction. To unravel the molecular, cellular, and circuit processes that underlie memory, her lab uses a multidisciplinary approach that focuses on mice and attempts to translate these basic findings to humans. Dr. Josselyn received the Innovations in Psychopharmacology Award from the Canadian College of Neuropsychopharmacology and the Efron Award from the American College of Neuropsychopharmacology. And she was also recently named a Fellow of the Royal Society of Canada. She's happy to take your questions after the talk, and it's my pleasure to welcome her.
Well, thanks so much, Steve, for that very kind introduction. And thanks for the organizers for the invitation. It's been really a fun meeting for me, getting to see the breadth of science, and particularly neuroscience in Texas. Who knew there were so many amazing universities and people doing such a diverse array of things? So it's really my pleasure to be here. I've learned a lot already. So what my lab is really interested in is understanding how the brain encodes information. We think this is a really fundamental goal, not only for understanding what it is that these brains do, but we think that understanding how information is encoded, stored, and used in the brain, that this is really important for mental health and also many other diseases. You can imagine something like autism or even something like Alzheimer's disease can be thought of as really a breakdown of this real fundamental process of how we encode and use information in the brain. And we've heard a number of talks already today about memory and how we form memories, how we use memories. And what my lab is looking at is memory at the sort of cell ensemble level. So we're going to call those bits of the brain, those cells in the brain that encode and store a memory, I'm going to refer to it as an engram. And this is a very old word, engram, and I'm going to tell you a little bit about the history of it. So back in 2017, my colleagues Paul Franklin, Stefan Kohler, and I wrote this really fun paper in the Journal of Neuroscience where we identified 7 of our heroes of the engram. So these are 7 scientists who came before us who we thought made real contributions to our understanding of how the brain encodes and stores information. And this was a really fun exercise for us because it allowed us to really dive back into the history and sort of look at the contributions and who was overlooked perhaps and who was, really celebrated in their times, and go back and read the history, because a lot of times for younger scientists, it's really, they overlook the history of something. And I think when you overlook the history of something, you're really doing yourself a disservice. So in my lab, we try and, dig up really old papers and read them and try and really understand the contributions of this. So back in 2017, when we were writing this article, we were joking that we picked these 7 luminaries: Richard Semon, Karl Lashley, Donald Hebb, Wilder Penfield, Brenda Milner, and James McConnell and Richard Thompson. And we said, well, look, these are 7 people that would be ineligible for a Nobel Prize. Most of them were, these are fairly historic figures, so many of them were already deceased at the time and therefore ineligible for a Nobel, and Brenda Milner was a woman. So of course, now the Nobel they've sort of become more enlightened. This past year, there were 2 female Nobel laureates for science. But hopefully we were changing the times. So one of the first engram heroes I'm going to talk about is Richard Semon. And he was a German scientist who's really interested in sort of evolutionary biology. And he was the person that originally coined the term engram. So he made this wonderful definition of an engram, which is very similar to what we nowadays call a memory trace, and he defined it as the enduring, though primary latent modification in the irritable substance produced by a stimulus. So that bit of the brain that is changed by a learning event. And I like this definition because it sort of says everything, but it doesn't really say anything in particular. So we don't really understand what it is, but we think it's sort of a really good shorthand and a really good organizing principle. He also coined this term called ekphory, which is equivalent to memory retrieval, and it is the influences which awaken an engram out of its latent state into one of manifested activity. So this engram, this sort of memory trace, lies dormant until you need it, then you reactivate it and you recall this memory. So I'm going to move on to our next hero of the engram, and this is Karl Lashley, who many of you may have heard of if you took Psych 101. And Karl Lashley, of course, is an American scientist who spent over 30 years of his very illustrious career searching for an engram in the brain. So what he did is he said, okay, I really want to localize that bit of the brain that's important in storing a memory. So he would train rats on a maze such as this. He would train the rat to find this goal, which was probably had a bit of cheese in it. And after training this rat very well, he would then went in and lesioned out bits of the cortex, trying to find where in the brain this memory was stored. So if he tested the rat again, the rat wouldn't remember, and he would say, aha, that bit of the brain that I had removed is the place that stored this memory. This is the location of the engram. And after, as I mentioned, about 30 years of work, he wrote this very influential paper where he summarized his findings. And basically, he concluded that the engrams seemed to be everywhere in the cortex, because if you made a lesion big enough, the rat would certainly forget, but nowhere specifically. So he could never pinpoint that area of the brain that stored this engram. So when I started my lab, I was really inspired by these people that went before me, and I wanted to continue the search for the engram, but I wanted to change it up a little bit. I figured that maybe one of the reasons why Lashley couldn't find the engram was because he was using this fairly complex task. So I wanted to break it down and start very simply. So I wanted to use a very simple task called auditory fear conditioning, and in this task, I take a mouse and I give it a single pairing of a tone and a shock, one single type of training. Then at any time later, I can give the animal, represent the tone that was previously paired with shock in a new environment. And if the animal remembers the last time I heard this shock, the last time I heard this tone, I received a foot shock, this mouse will display a number of species-specific responses, including this response called freezing. So when a mouse is afraid, it displays this active behavior where it adopts this crouched motionless posture, and it's freezing. So we can always use this index of how much this mouse is freezing when we replay this tone as an index of their memory. So I use this task because it's very quick, it's very simple, one trial learning. We can observe it in the lab very easily. And also because some real pioneers in the field told us where to look in the brain for this type of memory. So I did my postdoc. This is Mike Davis. I did a postdoc with Mike Davis. This is Joe Ledoux, a real luminary in the field, and Michael Fanslow. And these really eminent scientists, as well as some others who refused to have their picture taken with me, have really showed us where in the brain to look for this type of memory. So this is a mouse brain. This is the coronal, so an ear-to-ear section. And they showed us that this area of the brain right here, the lateral nucleus of the amygdala, is really key for this type of conditioned fear memory. And when we looked in the literature, we saw that although most lateral amygdala principal neurons, so these big excitatory neurons, seem to be correctly wired to become part of a memory trace, that a number of authors showed us that only a relatively small portion, around 20%, give or take, seem to be necessary to encode any one given fear memory. And this is really similar to what Ray Dolan's lab has published, showing a stable, sparse fear memory trace in the human amygdala. So we asked a very simple question. If fear memories are sparsely encoded in the lateral amygdala, why is this one neuron and not its neighbor somehow chosen to become part of a particular engram? So in our studies, what we did is we tried to look at how one neuron is chosen over another neuron to become part of this engram that supports a conditioned fear memory. So this is sort of the take-home message of our findings, is that we think that all eligible lateral amygdala neurons seem to compete against one another for allocation or recruitment into a particular engram, and that these neurons that win the competition, that get to be part of this engram, are neurons with the highest level of the transcription factor CREB. So these neurons with increased CREB function win this competition. Now, what is CREB? Well, it's our favorite transcription factor. And we became very interested in CREB because of studies in the 90s in Drosophila by Tim Tully and Jerry Yin, and in aplesia, of course, by Eric Kandel. This is me, that's my daughter Charlotte at some event with Eric Kandel. So these people using invertebrate models have shown us that CREB is really important in memory. So what my lab wanted to do was to see what would happen if we manipulated CREB levels in individual lateral amygdala neurons. So to do this, we're going to use a viral vector that overexpresses CREB. We infuse it into the lateral amygdala of mice, and it's going to randomly infect 10% of excitatory neurons. And when we do this, we're going to look at two things. First, the memory, again, the percentage of time that these mice spend freezing to the tone. And we're going to look at what this does to the engram architecture. So how does the memory trace look after we've overexpressed CREB in a random percentage of cells? And to do this, we're going to look at neurons that are active after memory recall. And we know that a neuron that's active after memory recall shows an increased level of ARC, which is an activity-dependent immediate early gene. So we're going to test the mice, and then immediately after the test, we're going to slice up the brains and look for neurons that have high levels of this immediate early gene ARC, the mRNA. So these studies were done by Jin-Hee Han, who was a postdoc in my lab, now has his own lab, and Stephen Kushner, who also has his own lab at Erasmus. And what they did is they found that when they increased CREB levels in a small portion of random cells in the lateral amygdala of mice, that these mice subsequently showed higher memory than their GFP, so their control counterparts. So the increasing CREB in a small percentage of neurons increased memory. And when we looked at the overall engram architecture using our ARC mRNA as a signature of an active neuron, we found that these neurons that were expressing more CREB were 3 times more likely than their non-infected neighbors to be recruited into the engram. So this suggested that not only were we enhancing the memory, we were changing the shape of the engram. So then they asked, well, what happens if we do the exact opposite experiment? This is what happens if we increase CREB in a random population of cells, but what if we decrease CREB in a random small population of cells? What happens to memory? So to do this, we're going to use another viral vector approach where we express a dominant negative, so like a CREB antagonist, and we're going to infect a small population of random cells, and we hypothesized that nothing at all would happen to memory because the remaining number of non-infected cells, non-manipulated cells, would be more than enough to house a memory. And this is exactly what we found. So first off, memory was normal in these mice. And second, when we looked at the architecture of the engram, we found that cells that we had infected, so cells with lower levels of CREB, were now 12 times less likely to be members of the engram supporting the sphere memory than their non-infected neighbors, as if the memory was being actively excluded from these neurons. So together, these studies led us to conclude that neurons compete against one another for allocation to an engram, and those neurons that win the competition are the neurons with increased levels of CREB. But this is very correlational. So we wondered, well, if we think that these neurons with increased levels of CREB that win this competition are really important in supporting this engram, the experiment what we really want to do is to target these neurons just after a mouse has learned a memory and see what happens to the memory. If these cells are really important, we would suspect that memory would go away. And this is exactly the experiment we think that Karl Lashley would have done if the tools were available to him at the time. So this is what we did. We took advantage of diphtheria toxin, which we've already heard about, as a way to kill specific cells. So as before, we have a group of mice where we're going to overexpress CREB in random cells, or we're going to overexpress just a marker, GFP. We're going to train and test the mice. And before we've done any killing, we're just going to check the memory of these mice. And memory is good in both groups. But then, in one group of mice, we're going to selectively kill just these neurons we've overinfected with CREB, and we're going to test the mice again. In the other group, we're going to kill just, again, a random population of cells that are expressing GFP. So when we do that, we're going to test these neurons. We're going to kill these neurons we think are really important to the memory, and then we're going to test the mice again. And when we do that, we see that the memory is decreased if we kill the cells that were overexpressing CREB and not in another group of mice where we kill the same number of cells, but cells that were not necessarily part of the engram. So we thought this was fairly good evidence that these neurons overexpressing CREB were really important in the memory. But then Asa Rasheen and Chen Yan joined my lab, and they said, killing cells is so permanent. Is there a way we can test their importance in the memory trace without killing them? So for this, we took advantage of the very cool tools offered by the people who invented optogenetics. Here is Karl Lashley, Ed Boyden, and Peter Hagerman. And just this past year, they won a Gairdner Award, which is like the Canadian answer to the Nobel Awards. We call it the Golden Globes of Science because it's a fun dinner and everybody drinks too much. So based on the wonderful tools offered by these guys, we decided to use optogenetics. So as you probably have heard about already today, we're going to use an optogenetic construct called halorodopsin, and when you shine a light on halorodopsin, you get an influx of chloride ions, and this inhibits the cells. So what we want to do in this experiment is we're going to have CREB. We think cells are that the memory is allocated to those neurons overexpressing CREB, and we're going to test the memory first when we inactivate the cells, and then when we don't inactivate the cells. So we're really going to test to see whether these cells overexpressing CREB are key in the memory or not. Of course, we're going to do this memory test counterbalanced, so some mice are tested with the cells normal, and then we're going to inhibit the cells. So only when we inhibit these cells overexpressing CREB do we see a memory deficit. If we inhibit another population of cells, we don't see this deficit. So we saw this once with halorodopsin, and all of these optogenetic constructs have pluses and minuses. So we tried it again with another type of inhibitory opsin, this time that's activated by blue light, and we see the exact same results. So from this, we concluded that lateral amygdala neurons overexpressing CREB at the time of training are necessary or somehow indispensable for the subsequent expression of that memory. So this is my daughter Charlotte a few years ago, and she's wearing a t-shirt that the lab bought for her, and she's asking this really important question. Well, why are these cells overexpressing CREB so special? What is it about them that makes them more likely to be recruited into an engram or a memory trace? Now, as I mentioned, CREB is a transcription factor, and it's been implicated in a number of different processes. But based on the work from Rob Malenka's lab at Stanford, we decided to look at neuronal excitability. Now, his lab showed that neurons with increased levels of CREB are more excitable than their neighboring cells. So this is Adelaide Yu and Valentina Mercaldo, two postdocs in the lab, and they said, well, if we think that overexpressing CREB serves to increase excitability, and that's the key to the effect that we're seeing in allocation, can we just artificially increase excitability of a neuron and bypass CREB, take out the middleman, see the exact same results, so enhanced memory, and these neurons preferentially recruited to the memory trace without directly manipulating CREB? And so they did this using first a dominant negative potassium channel, second using optogenetic tricks, and third using chemogenetic tricks. This is Brian Roth from UNC, and he made these fantastic chemogenetic tools. And using all of these tools, they came to the same conclusion, that an allocation to an engram seems to be a competitive process between eligible principal neurons in the LA. The outcome of this competition really seems to be based on excitability at the time of training. And of course, one of the way endogenously neurons show increased excitability is by overexpressing CREB. So we've shown that allocation to an engram seems to be important in the sphere conditioning memory in the lateral amygdala. But other groups, as well as our group, have also shown this seems to be a process that seems to be seen across the brain in a couple of different tasks. So we've shown it for fear conditioning and also for rewarding memory in the lateral amygdala. Alcino Silva's lab, who was my second postdoctoral advisor, has shown this in the retrosplenial cortex. We have some groups showing this in the piriform cortex for an odor-related memory, in the dentate gyrus for different types of memory, and also in the CA1 of the hippocampus. And a really cool finding from Albert Lee's lab at Janelia showed that hippocampal place fields seem to emerge upon single-cell manipulation of excitability during behavior. Now, place cells and engram cells, they may have similarities and may have differences, but it's really cool that he can manipulate excitability of a single cell, make a silent cell turn into a place cell. So we're wondering how generalized this phenomenon is, where exactly is it important, why is it important? And so these studies are ongoing. So this is a very hardworking postdoc in my lab, and his name is Asim Rashid. And he said, well, what's the function of allocation? Why would we need it? So in our labs, in our studies, what we do is we train mice and we look at what happens. And usually we manipulate CREB beforehand. But in one study, he looked at CREB after just manipulating training. He didn't do any genetic trick, just a normal mouse trained it. And after training, he found an increase in the levels of phosphorylated or activated CREB in the time following training. So 1.5 and 6 hours after training, he found increases in phosphorylated CREB or activated CREB, but he didn't see this in the hours after training. So this made us wonder that what are we really doing when we artificially manipulate CREB in the amygdala? Was maybe what we're doing is mimicking an endogenous process of previous learning. So our model came like this. So say a certain training event one, so a fear conditioning task, that would lead to increase in excitability, an increase in CREB function in a population of 1 cells in the lateral amygdala. And this increase would last, say, about 6 hours, such that if a second training event, a related training event, happened within this 6-hour time period, these neurons that were activated by the first event are still more active, and they would be allocated to the second event. So a similar population of neurons would be co-allocated to these 2 events, and this would link memories. But CREB is very cool. Once it's turned on for a while, it has a capacity to turn itself off, and when it turns itself off, there's an undershoot of excitability, like a refractory period, such that if a second training event happened during this refractory period, the second memory would be allocated to a distinct population of cells. This would mean we're going to call this disallocation, and the 2 memories would be separated. Event 1 would be encoded by one population of cells. Event 2 would be encoded by an entirely different population of cells. So we tried to check out this process by collaborating with my very close personal friend, Karl Lashley. There's my selfie with Karl Lashley, and some very talented people in his lab, Charu and Sun-Yoon especially. And what we did was make a new viral vector that expressed both channel rhodopsin and halorhodopsin. So this gave us the possibility to both excite cells by turning on channel rhodopsin or inhibit the same population of cells that we've infected simply by shining different wavelengths of light. So in this experiment, what we're going to do is really get to the heart of this allocation process. So first, we're going to infuse this vector that has both channel rhodopsin and halorhodopsin, and we're going to allocate cells to the memory trace by shining the blue light to excite the cells. We're going to fear condition the mice. Now, how do we know that these cells are involved in the memory trace? Well, during the test, we're going to simply shine the red light to inhibit these cells and see what happens to the memory. So when we inhibit these cells during the test, we see a memory decrease. And we know this is specific for these cells because if we inhibit just a random population of cells that we didn't allocate, didn't excite before the training period, we see no effect at all. So using this really cool vector allowed us to really gain control of the activity of these neurons. So what we did in this experiment was to take the principal cells that we've infected, and we're going to manipulate them optogenetically, but we also know there's other really important members of the lateral amygdala that could be important in this process, and we keyed in on these parvalbumin positive interneurons, so these GABA interneurons, and they populate the lateral amygdala and they innervate it very widely. So we're going to optogenetically control these principal neurons, and we're going to chemogenetically control these parvalbumin positive neurons. And the results of these very complex experiments led us to conclude that indeed 2 events separated by 6 hours do seem to be co-allocated to the same or similar population of cells, linking the memories. The same 2 events, if we separate them by 24 hours, the memories are not linked, they are separated, and we think there's a disallocation process going on. So if we want to sort of show this in terms of neurons, we think that allocation is a time-limited winner-take-all process that serves to either integrate or separate memories based on temporal context. So say event 1, we have A, B, C, and D neurons that are expressing or holding this representation. If event 2 comes along, it's pretty similar. It's in a small time window thereafter. We think maybe B, C, D, and E neurons would be representing this memory. And if event 3 happens a long time later, it would be represented in an entirely distinct population of neurons. So that if we were to recall event 1, we would automatically start to recall event 2. We would therefore link these 2 events in our memories via a pattern completion process. But we would keep these two memories distinct if event 3 were encoded by an entirely distinct population of neurons. So these are our studies that look at this process of linking memories in mice, but we're really excited. There's also been some amazing work in humans looking at this linking process using humans in fMRI work. I'm then going to switch channels a little bit and tell you about some work that a graduate student in my lab, Chen Yan, did. And Chen was really interested in this allocation process, but all the things that we're doing so far in the lab are very, very invasive, and they're not physiological. We can manipulate the activity of different neurons, but does this process happen endogenously? So Chen was really inspired, as I think we all were, by Mark Schnitzer's talk yesterday, and the way that he can see neurons fire in real time. So he made this mini endoscope where you can place it in the brain and actually see neurons firing in real time. And Chen was so excited, he made his own. So this is a mini endoscope. Chen's name is Chen. We call this a chendascope. So he made his own chendascope. And the plans and everything are freely available. Here's his chendascope. This is a Canadian $2 bill, Canadian $2 coin. And together with a bunch of other people in the lab, and Dave Reddish and Nathan Insel, as collaborators to help us with the data analysis, we have made this chendascope and are trying to look at whether this process of allocation is occurring normally in mice. So this is just, these are cells that are, we can see labeled with this calcium-dependent label, and you can see the activity of different neurons firing. This is our very first iteration of this chendascope, and as you can see here, one really important thing is that there's a bit of tinfoil there, and that makes sure that we are actually recording the calcium transients of the mouse brain rather than aliens. So now we're going to use this chendascope to try and look at this process of allocation. But it's difficult to see down in the amygdala. So we're going to start with an area that's easier, closer to the surface. And using this chendascope, we do see evidence of allocation in the dorsal hippocampus. So those neurons that tend to be more active right before the training event do seem to be allocated into a hippocampal-based memory. We're still looking at this in terms of the amygdala. Those studies are ongoing. But we've also used this to look at memory dysfunction in Alzheimer's, or maybe I should now start calling it Fisher's disease. I've been learning a lot about Alzheimer's disease. And what we're trying to do is really get at the core of the memory dysfunction. How is the circuit not working properly in mouse models of Alzheimer's disease? And I'm just going to finish up by telling you some work of Emily Kramer's. She's a graduate student in the lab. And what she wanted to do is to see, well, maybe the form of the amygdala engram is important in its function. So what she wants to do is to look at the entire amygdala from the very beginning to the very end of the amygdala and to see how it's structured, the engram. What's the architecture look like? And okay, there we have a spin of what she sees. So usually when you do these sorts of studies, you take 50-micron sections of a mouse brain from the beginning to the end, and you take every few sections and you try and reprocess it and put it together in a 3D image. But we're missing a lot of tissue. We're missing a lot of information using it this way. So many of you may know, this is Mariah Carey. Mariah Carey, for any of you who don't know Netflix, that she was a singer way back. I think she still is a singer, I'm not sure. Anyway, back in 2009, she sang this song called Obsessed. And in this song, she said, "I can see right through you, like you're bathing in Windex." Now, for my money, I think Mariah Carey inspired all these new tissue clearing techniques. So these tissue clearing techniques allow you to see right through the brain like it's bathing in Windex. So taking advantage of these very cool techniques, we can now see the entire amygdala in 3D. So what Emily has done is use this technique to try and see the 3D nature of an engram in the amygdala. So we can use the precise coordinates now and really make sure we're looking at exactly the lateral amygdala by comparing it to the Allen Brain Atlas. And as you can see here, the colors are cells that were active following a memory recall event versus another control group of mice. And she's really looking at, are the cells, are they chunked together? Are they totally random? Are they chunked together? Are there real nodal points? What happens during different things? What happens after extinction? And some really important questions just by looking at exactly the architecture of the engram in the lateral amygdala. So in our studies, we really want to find out how the brains of mice work so we can really understand how the brains of people work. So with that, I just want to thank my main collaborator, my husband, Paul Franklin. Here he is with my daughter again, Charlotte. She usually features in most of my slides. Thank my funding organizations, but importantly, thank the really good people of the Josselyn Franklin Lab and all the hard work that they do. The people in my lab stopped coming out for the annual picture day, so now we have this annual selfie day. So every April 24th at 10:30, because who doesn't like a selfie, everyone has to take a picture of exactly what they're doing. And every year we get more and more creative. My hope is that they put some of this creativity into not only their selfies, but also into their science. So thank you so much for your attention.
Would you like to answer any
Sure,
questions?
absolutely.
We have some time for one or two questions if the audience would like. There we go.
It's a great talk, Sheena. I was wondering, what do you think is the kind of mechanism for the increased excitability in the CREB cells? Because if you kind of think about neurons getting recruited to an engram, you'd almost think that those neurons that were already in an engram are not available to be recruited, but then you would sort of, with the traditional ideas in the field, you would think that those would be the cells with potentiated synapses. So I know it's probably like intrinsic excitability, but it's like a baseline level of excitability, and the other ones are maybe more sharpened between their baseline and their evoked excitability, or is that the idea?
Yeah, absolutely. So we're still trying to understand the mechanism, especially with endogenous. I mean, we can manipulate things, but really what's happening endogenously. So we're thinking exactly along those same lines, that for whatever reason, when you start at sort of time zero, some cells are just more active, and we sort of say that's maybe waves of calcium going through. And then the cells that become more active with the memory, then they're also likely to be encoded in the next memory, if it happens within a certain period of time. So yeah, recent activity, recent learning primes you to become involved in the next bit, then you drop out of the circuit for a while, a new population comes up, and they become encoded. And we think this is maybe how you get sort of something like knowledge, where it's not just one thing you learn a whole basket of things, and they all pop up together.
And do you think there's any interaction with what's going on during sleep?
Absolutely, absolutely. So it didn't escape our attention that the difference between our 6-hour time point and our 24-hour time point is a sleep cycle. So we think that sleep might reset the schedule. It might reset everybody back to a baseline level so they're fresh for tomorrow. But we're still looking at those sorts of questions. Absolutely.
So thank you so much, Sheena, for such a wonderful talk, and for creating such a vivid engram for us at the end of the meeting. It's wonderful. So thank you very much, Sheena.